today we are going to discuss about reproduction in organisms. First, definition of the reproduction. The ability of an organism to produce new individuals of the same species is known as reproduction. So here every organism, it produces new organisms, new daughter one, young ones or offspring. Usually this offspring, again they produces new offspring. Because of this reproduction ability, what happens? So continuity will be there. So main important purpose of reproduction, to continue the species generation after generation. Now lifespan, definition of the lifespan, the period from birth to natural death of an organism is known as lifespan. So here from birth to natural death, usually lifespan, it is not constant for all living beings, that may be plants or animals. So lifespan, it is not correlated with size of the organism and the complexity of the organism. So it is not correlated with size of an organism and as well as structural complexity of the organisms. So for example, if you will see crow and parrots, so almost all size point of view will be same size, but if you will see the crow lifespan will be 15 years, but whereas parrot lifespan is 140 years. So here almost all, so size almost all will be same, but lifespan it will be very more difference will be there for both the organisms. As well as if we will see here mango plant and a peepal tree. So here mango and peepal, both the plants lifespan will be more difference. So here mango tree has very lesser or shorter lifespan than peepal tree. Now at present we are going to discuss about different types of plants and different types of animals, their lifespan. Now we are going to study about lifespan of different plants. First, rice plant. Rice plant lifespan 3 to 4 months. Second plant, rose plant. Rose plant lifespan, it will be around 5 to 7 years. Third, mango plant lifespan, if you will observe, 200 years. Banyan tree, it is average 200 to 300 years. Peepal tree lifespan, if you will see, it will be maximum 2000 to 3000 years. 𝗧𝗦𝗔𝗧 plant, 𝗧𝗦, it is a 𝗧𝗦𝗔𝗧 𝗚𝗏𝗏 plant, gymnosperm, tallest gymnosperm, lifespan will be 3000 to 4000 years. Macrogamy, here one more plant is there here, macrogamy. It is a gymnosperm, 10,000 to 12,000 years. Like this, different plants, if you will observe lifespan, months, rice plant 3 to 4 months, Macrogamy gymnosperm 10,000 to 12,000 years. If you will observe, so few plants morphologically size will be same height, so here but lifespan it will be differ. So mango plant lifespan 200 years, but if you will see people tree 2000 to 3000 years. So finally here what we can understand based on the chart, lifespan of the organism, it is not constant, it depends upon the type of plant, it changes. So lifespan mainly it is under control of habitat and internal morphology and external climatic conditions. All these factors responsible for lifespan of the different types of plants. So as well as if you will see here, some plants are there like ephemerals. Example tribulus. Ephemeral example tribulus. This plant it completes life cycle. So here few weeks to few months. So shortest lifespan you can observe. So here such type of plants also there, ephemerals. So here as well as if you will see banana plant. So banana plant lifespan will be 25 years. So like that if you will see the different plants lifespan will be different years. Now at present we are going to study about few animals and their average lifespan and the average lifespan of the different types of animals. So first one insect, mayfly lifespan will be only 1 day. Butterfly lifespan will be around 1 to 2 weeks. Average lifespan of butterfly. Fruit fly, average lifespan if you will observe will be 30 days. Crow lifespan it is 15 years. Cow lifespan if you will observe 22 years. Dog lifespan 20 to 30 years. Crocodile lifespan 60 years. Horse lifespan 60 years. Elephant lifespan 60 to 90 years. Tortoise lifespan 100 to 150 years. Parrot lifespan, average maximum lifespan 140 years. Now if we will observe this chart, we can understand minimum lifespan of the animals that starts with 1 day to maximum years 150 years. So here the mayfly only the lifespan will be 1 day. So butterfly 1 to 2 weeks, Like that, if you will see here, different animals having different lifespan periods. So lifespan, so it is not constant among different types of animals. So lifespan of the plants, it is not constant years. So as well as animals lifespan also, it is not constant. So that is different animals lifespan will be different years, different days, different months. Reproduction mainly divided into two types. First type of reproduction, that is asexual reproduction. Second type of reproduction, sexual reproduction. In the asexual along with different types of asexual reproductive methods, there is one vegetative reproduction also there. Vegetative reproduction also a kind of asexual reproduction. At present, we are going to discuss about asexual and sexual reproductive methods. So differences. And mainly in asexual reproduction, only single parent is involved. That is uniparental type of reproduction is known as asexual reproduction. So whereas in sexual reproduction, it may be here single organism may involve or two organisms also may be involved. So that, for example, in bisexual flower, for example, a plant is there, that plant it is producing bisexual flowers. Pollen grains transfers from anther to stigma of the same flower. So pollination takes place, fertilization takes place. That is sexual reproduction. But here, there, in that case, so only single plant is involved. So sexual reproduction may be one or two organisms may involve in sexual reproduction. Asexual reproduction, there is no involvement of gametes. Gametic involvement does not takes place in asexual reproduction. Whereas in sexual reproduction, gametic production will be there. Fusion of gametes also takes place. So asexual reproduction, it is very fast process. So very less time required for production of offspring or new generation or young ones. But whereas sexual reproduction, it is very slower process. It takes very slowly in different types of organisms, that may be plants or in animals. So asexual reproduction usually, so offspring or young ones, they are resembles like parents. They are exactly appears like parents due to involvement of only single organism. But whereas in sexual reproduction, if you will observe, so organisms or offspring, they are not exactly identical to parents, because whatever organism produced because of sexual reproduction, so that organism it is formed because of fusion of gametes. Male and female gametes are fused and it leads to formation of zygote. That zygote develops into the young ones. So because of gametic fusion, offspring, they are not 100% identical to parents, but few characters. So we can see either male or female parent, a few characters you can see, or combination characters, so we can see, or sometimes 3-4 generations before generations characters also sometimes you can see in the progeny, whatever organisms formed because of sexual reproduction. So here usually asexual reproduction, whatever organism is formed 100% identical to parent, that's why the type of offspring known as clones. The organism which is formed asexually, such type of organisms are known as clones. Usually clones are identical to parent. So whereas here clones are not formed in the sexual reproduction. Because of gametic fusion, new organism is formed with new characters. Usually asexual reproduction you can see in primitive organisms and as well as in plants also you can see asexual reproduction. Sexual reproduction we can see in the primitive organisms and as well as in higher organisms. At present, we are going to discuss about asexual reproduction, different methods, what type of methods you can find in different organisms, how different organisms reproduces new offspring because of asexual reproduction. So all different kinds of asexual reproductive methods at present we are going to study. Methods of asexual reproduction. Different types of methods. First method, fission. Second method, budding. Third method, fragmentation. Gemmules. 𝗳𝗧𝗃 or 𝗳𝗦. Conidiospores. Sporangiospores. Aplanospores. Chlamydospores. Actinids. Next, zoospores, statospores, endospores, Harmagonia, amylum stars. Like this, at present we are going to discuss about different types of asexual reproductive methods. So at present, total different types of organisms, they exhibits different types of asexual reproductive methods. Sometimes one single organism also exhibits 2-3 different types of asexual reproductive methods. Mainly asexual reproductive methods takes place in different types of organisms to produce offspring within short span of time, or young ones produced more in number in asexual reproductive methods when you compare with sexual reproductive methods. So here the first method, fission. Second method is budding. Third method, fragmentation. Fourth method, gemmules. Fifth method, 𝗳𝗃𝗃. So inside the 𝗳𝗃𝗃 you can find a 𝗳𝗦. Sixth method, conidiospores. Seventh, sporangiospores. Eighth method, aplanospores. Ninth, chlamydospores. 10th, actinids. 11th, zoospores. 12th method, statospores. 13th method, endospores. 14, Harmagonia. 15, amylum stars. All these different types of methods exhibited by the different organisms. First sexual reproductive method, fission. The fission type of sexual reproductive method we can see in many unicellular organisms. In this method, usually was single organism, it produces maybe two organisms, maybe many organisms. Fission method of reproduction we can see mainly in primitive organisms like protozoans and moneyrons. Fission method of reproduction again divided into two types, that is binary fission . Second method, multiple fission. Fission method of asexual reproductive method divided into two types, binary fission, multiple fission. At present we are discussing about binary fission method of reproduction. The binary fission first method of reproduction, first method of binary fission, that is irregular binary fission, that is otherwise known as simple binary fission. In this type of binary fission, what happens is a single organism, that is unicellular amoeba, irregularly, there is no particular direction for division. So at the time of simple binary fission, this total nucleus of the organism, that is amoeba, elongates like this. Then finally what happens, because of development of a constriction, it leads to formation of two daughter organisms. There is no particular direction for division. It may divide in any plane. So finally it leads to formation of two daughter cells. This type of binary fission is known as simple binary fission. Second type of binary fission, longitudinal binary fission. In this method, longitudinal binary fission, so usually organism, it divides longitudinal to its axis, so that can be seen in euglena. In euglena, total organism, it divides longitudinally into two equal halves. The type of binary fission is known as longitudinal binary fission. Transverse binary fission, we can find in paramecium. So here in the paramecium, at the time of binary fission, what happens? So it divides transversely. It leads to formation of two daughter cells. So this type of binary fission is known as transverse binary fission. Fourth type of binary fission, that is oblique binary fission. In this method, what happens? Organism, that is Serratium, example for oblique binary fission. Serratium, it is a dinoflagellate. At the time of binary fission, obliquely it divides into two daughter cells. The type of binary fission is known as oblique binary fission. Like this, different types of binary fissions methods we can find in different organisms. At present we are going to discuss about, in the fission topic, multiple fission. It is second type of fission method of asexual reproduction. In the fission method, at present we are discussing about multiple fission. In this method, inside the organism, protoplasm, next nucleus, divides many times and forms many daughter organisms within the parent cell. So because of multiple fission, at a time from single parent organism, many daughter organisms comes outside because of rupture of the cell wall or cell membrane of the organism. So in the multiple fission, at a time more number of organisms are produced. But whereas in binary fission, single parent cell divides into two daughter cells. But multiple fission, at a time many organisms are produced from single organism that can be seen in amoeba and plasmodium. In these organisms, we can observe multiple fission method of reproduction. So these multiple fission and binary fission both are two types of fission method of reproduction. These organisms offspring, they are identical to parent cell. At present, the second type of reproduction that we are going to discuss about budding method of reproduction. Second method of asexual reproduction, budding. Budding method of asexual reproduction can be seen in different types of organisms. So first example is yeast. It is best example for unicellular fungus, as well as the cnidarians like hydra also exhibits a budding method of reproduction. Present we will discuss about budding method of reproduction in the yeast cell. Yeast cell, it is a best example for unicellular fungus. Chitinous cell wall we can find. True nucleus, it is eukaryotic. That's why we can find true nucleus, that is membrane bound nucleus we can find. Cytoplasm we can find around the nucleus. At the time of budding method of reproduction, at one particular place, cell wall, it becomes very thin. Wherever that organism, it want to produce birds. So at that particular place, what happens, cell wall becomes very thin. So as thin constriction, cell wall, it becomes very thin at one particular place. There what happens, it bulges out and it produces a bird. After development of the bird, here what happens, whatever cell wall found between, this is parent cell, it is bird. Whatever cell wall present between bird and parent cell, that cell wall disintegrates or disappears. Then slowly what happens, cytoplasm of the parent cell pushed into the bird. Then this nucleus divides mitotically and produces two daughter nuclei. In any one nucleus, here two nuclei are formed in the parent cell. Any one single nucleus that is pushed into the bird. Then here constriction, it will form between bird and parent cell. This bird detaches from the parent cell. Then whenever it falls on the suitable substratum, it enlarges its size and acts like new organism. The type of reproduction we can find in yeast cells. But here budding method can be seen in hydra also. In the hydra, usually you can find tentacles. At the time of budding method, what happens? A small bird, lateral bird develops. This bird also develops tentacles. So this bird after maturity detaches from the parent hydra and acts like new hydra. So these methods, especially the hydra yeast, whatever budding method found in these organisms, usually these birds, they are formed outside the parent organism. So if you observe here clearly, it is parent organism, this is bird. This bird, it is produced outside the organism. That's why it appears like a spore. That's why exospore is formed. These type of spores are known as exospores. Bird, it is equivalent to a small spore. But exogenously, these birds are formed. So birds are exogenous spores, exogenously produced from the parent organism. This type of reproduction also we can see in different organisms. Present we are going to discuss about third method, fragmentation. Fragmentation method of asexual reproduction can be seen in different organisms. We can find in majority of the algae and as well as in bryophytes also you can find. In general, the fragmentation method of reproduction. So here a parent organism, so because of big animals or because of environmental conditions, if the organism, if it breaks into the small fragments or pieces, each and every fragment develops into the new individual organism. For example, in the algae, in the Spirogyra algae, it is filamentous chlorophyceae green algae. So here in the Spirogyra, in this alga, because of water current or animals, if this filament, if it breaks into the fragments, each and every fragment develops into the new organisms. It is a simple method. So this type of asexual reproduction also we can find in different organisms. Fourth type of asexual reproductive method, gemmules. Gemmules are internal birds. They are produced in different organisms, especially in the sponges. In the sponges, internal birds, multicellular internal birds we can find in sponges. These internal birds which are formed in the sponges known as gemmules. These gemmules, so they comes outside, they act like new sponge. Gemme, it is also a type of asexual reproductive method. In the bryophytes, especially in the liverworts, Hepaticapsida class plants, they develops on the gametophyte gemma cups. In this gemma cups, small gemma develops. These gemma are multicellular autotrophic green color photosynthetic structures. These gemma detaches from the parent gametophytic plant. Whenever this gemma falls on the suitable substratum, develops into the new gametophytic plant. So gemma we can find in many bryophyte plants. It is also a type of asexual reproductive methods. Conidiospores we can find in Penicillium and Aspergillus fungus. So these type of spores, usually these spores, they are formed in majority of the fungi. It is a fungus, mycelium is there, hypha, a single hypha is there, At the time of production of conidiospores, it develops a vertical structure. This is known as conidiophore. Apex of the conidiophore develops special structures. These are known as rami. At apex of the rami, they develops again some more structures. These are known as medullae. At the apex of medullae, a flask-shaped structures will develop like this. This is known as sterigmata or phyalides. Apex of sterigmata, small spores will form. These spores are known as conidiospores. These conidiospores, they are produced basipetal order. Apex you can find matured conidiospore, at base immature or developing conidiospore we can find. So conidiospores also a type of asexual spores. These conidiospores detaches from the parent plant. Whenever it falls on the suitable substratum, it germinates, develops into the new hypha, new fungus. Sporangiospores, type of asexual reproductive method, we can find in the mucor fungus. Sporangiospores, they are formed in specialized structure that is known as sporangium. In this method, it is a fungus is there, fungus hypha. It produces a vertical filament stalk that is known as sporangiophore. Sporangiophore, phore means stalk. Apex of the sporangiophore, it swells and forms a special sac-like structure. This is known as sporangium. Inside the sporangium, many spores are formed. These spores are known as sporangiospores. Usually, after rupture of the sporangial wall, sporangiospores are liberated. After falling on the suitable substratum, develops into the new fungus. Usually here, these spores are very small, minute, microscopic, light in weight spores. Such type of spores, they are formed inside the sporangium, many number. Each and every spore, after falling on the suitable substratum, develops into the new fungus. Aplanospores can find in majority of the algae. Aplanospores, these are non-motile spores. They are formed in many algae. Usually aplanospores, these resembles like zoospores only, but zoospores consist of flagella, but whereas aplanospores, they do not consist flagella. So these non-motile spores which are formed in algae known as aplanospores. Ninth type of spores, chlamydospores. Usually chlamydospores we can find in Rhizopus fungus. These chlamydospores, how these are formed, we will study. In Rhizopus hypha, it consists of cells. So wherever plant or wherever this Rhizopus want to produce rhizospore, chlamydospores, that cell wall becomes more thick. It is chlamydospore. Here also a chlamydospore, it is developing during unfavorable conditions. To tide over unfavorable conditions, only these chlamydospores are formed. So here chlamydospore is formed, here also chlamydospore is formed. Between these two chlamydospores, a cell is there. Usually this cell, it becomes empty. So whenever favorable conditions appears, so these cells disintegrates, chlamydospores are liberated. These chlamydospores acts like develops into the new individual organism. Actinids we can find in 𝗧𝗦𝗔𝗧, 𝗧𝗏𝗺. In 𝗧𝗦𝗔𝗧, usually what happens, thick-walled spores are formed. These thick-walled spores usually formed during unfavorable conditions. So these thick-walled spores which are formed in 𝗧𝗦𝗔𝗧 are known as actinids. Whenever favorable conditions appears, so thick wall, it loses whatever coats present around it, then it acts like it develops into the new organism. Zoospores. So zoospores are motile spores. Usually these spores, they consist flagella. Depending upon number of flagella, zoospores are divided into different types. First type, biflagellate zoospores, biflagellated zoospores. Second, quadriflagellate or flagellated zoospores. Third type, multiflagellated zoospores. All zoospores are motile spores. Due to presence of flagella, they can move from one place to another in the water. Biflagellated zoospore, each spore consists two flagella. Example, Chlamydomonas. Quadriflagellated zoospore. These spores, they consist of four flagella. Example, Eulothrix alga. Multiflagellated zoospores, each spore, zoospore, it consists of more than four flagella. Such type of spores known as multiflagellated zoospores. Usually the multiflagellated zoospores you can find in the Edogonium alga. 12th type of spores, that is statospores. These specialized spores, they are formed mainly in the diatoms. These statospores, whenever favorable conditions appears, the spore, it acts like new organism. Next, 13th type, that is endospore. Endospores are formed in few bacterial cells to tide over unfavorable conditions. A spore-like structure is formed inside the bacterial cell. That's why these spores are known as endospores. These endospores, usually they consist of 3 layers. So different layers present around total endospores. So these endospores usually we can find in the many bacterial cells. Harmogonia. These are special fragments formed in cyanobacteria or blue-green algae. These fragments, they are surrounded by gelatinous sheath. Whenever total filament, that may be anabina, nostoc, so if it breaks into the fragments, so each and every harmogonium or fragment, it develops into the new organism. So these Hormogonia usually formed in blue-green algae or cyanobacteria. Last method, amylum stars. Amylum stars we can find in Chara alga. These amylum stars usually they appears like a star shape. These star-shaped amylum spores, they formed different nodes of the Chara. They store reserve food material. They detaches from the parent Chara alga whenever favorable conditions appears. It develops into the new Chara alga. So like this, amylum stars also we can find in Chara. This is also a type of asexual reproductive methods. Now at present we are going to discuss about vegetative reproduction. Vegetative reproduction also a kind of asexual reproduction. So definition of the vegetative reproduction. Here new individuals or offsprings, they are produced from vegetative parts like root, stem, leaves or branches. In this method, the production of offspring by vegetative parts like root, stem, leaves is known as vegetative reproduction. Whatever structure, whatever part of the plant helps to production of offspring, new young ones, that particular structure is known as vegetative propagule. Vegetative reproduction, two types: natural method, artificial method. Natural methods of vegetative reproduction. Here, in these methods, usually plants, by adopting all these different types of methods, they propagate, they produce new young ones to continue their progeny. All these are natural methods. So here the first different parts of the plant, they have ability to develop into the new plants. Roots, taproot and adventitious roots have the ability to produce new plants. So taproots, especially in the Murraya plant, that is curry leaf, new plants develops from the taproot. In few plants like dahlia, asparagus, hypomia, that is a sweet potato, in these plants adventitious roots also produces new plants. Underground stems also have the ability to produce new plants by vegetative propagation method. Here tubers, that is a tuberous stem, potato, rhizome, ginger and turmeric, bulb, allium cepa and allium sativum, carm, colocasia and amorphophallus. All these are underground stems also have the ability to produce new plants. Creeping stems. In few plants, usually if you observe, stem it creeps horizontal on the surface of the soil, and it also produces new plants directly from the creeping stem. So there are different types of creeping stems we can observe: runners, stolons, suckers and upsets. In the runners, grass plants and oxalis plants, so from whenever plant stem it creeps horizontal on the surface of the soil, from each and every node it develops roots. Unfortunately, if it breaks into the fragments, each and every fragment develops into the new plant. Stolons. Here stolons, example Nerium and jasmine. In this plant, stolons method of stolons helps to develop into the new plants. Suckers we can find in banana, pineapple, chrysanthemum indicum, that is in chamanthi. So suckers helps to development of the new plants. Offsets in especially in hydrophytes like Pistia, Eichhornia also known as water hyacinth. In these plants we can find new plants develops through offsets. Aerial shoot in sugarcane plant. So each and every node you can find a small bird in the sugarcane plant. So that bird has the ability to develop into the new plant. In few plants, leaves also have the ability to develop into the new plants. Begonia bryophyllum, at the leaf margins in bryophyllum, leaf margins, it produces small birds. Those birds are known as epiphyllous birds. Epiphyllous birds detaches from the parent bryophyllum leaf. If it falls on the suitable substratum, develops into the new bryophyllum plant. Bulbils, these are small birds develops aerially on the stem. These bulbils, they store food material, they detaches from the parent plant. After falling on the suitable substratum, develops into the new plant. In agave plant and Dioscorea plant, you can find bulbils. Here, bulbil different. Bulbils are smaller than bulbs. So all these are methods are natural methods. With the help of all these different methods, plants they propagate, they develops many new offsprings. Now at present we are going to discuss about artificial methods of vegetative propagation. So the artificial methods of vegetative propagation are divided into three different types: cutting, layering and grafting methods. Cutting method of reproduction. Here different parts of the plant like stem, leaf, root. So by cutting from the parent plant, if you will put in the soil, it develops into the new plant. So stem cutting, rose croton plant you can find. Leaf cutting, snake plant, sansevieria plant. Root cutting, lemon and tamarind plants you can find root cutting method. So the parts of the plant, different plants, for example rose plant, if you want to develop a new plant with the help of stem cutting, a simple method, just we have to cut a small branch or twig of the plant. That branch or twig, if you will put in the soil, it develops into the new plant. So like this, different parts of the plant have the ability to develop into the new plant. In the leaf, that is a example, snake plant, sansevieria plant. So leaf we have to cut horizontally. So after cutting, if you will put in the soil, it develops into the new plant. So like this, cutting method helps to development of the plants artificially. So root cutting we can find in the lemon plant. Lemon plant root, after cutting, if you will put in the soil, again it develops into the new plant. Next, layering method. Layering method, first definition of the layer, the branch which develops roots whenever present on the parent plant is known as layer. So layering method, two types: ground layering, air layering. Air layering also known as Guti method. Ground layering again divided into different types: tip layering, trench layering, serpentine layering. So in the tip layering method, for example in the tip layering method, if you will observe, in the blackberry plant, a root system is there. This branch we have to bend to the soil, toward the soil we have to bend. Then here what happens, this one we have to bend like this. This one, if you will put in the soil, it develops into the roots. This branch, after development of the roots, we have to detach from the parent plant. It develops into the new plant. The type of method you can find in the blackberry plant. Next, trench method of layering. Trench method of layering, what we can do here, in the walnut plant, from the stem, a weak stem we have to bend like this towards soil. Then horizontally we have to put in the soil. It develops roots like this. After development of the roots, we have to detach from the parent plant, develops into the new plant. Next, serpentine layering method can be seen in the clematis. Here roots are there like this. The serpentine method, a branch, weak branch, you have to bend at several places like this. Wherever it touches the soil, it develops roots. So if you break into the many fragments, each and every fragment, if it consists of roots, develops into the new plant. The type of method also we can find. Now at present we are discussing about air layering. Air layering, also known as 𝗬𝗽 method. Whenever branch it is strong, in strong branches usually we cannot bend towards the soil. Such type of branches usually we can develop the roots whenever present on the parent plant by air layering method. Usually in the air layering method, at present we are discussing about air layering. In the air layering method, the branch, we have to select a branch. These branches usually we cannot bend towards the soil because due to presence of hard tissues like xylem phloem tissues, complex tissues well developed. So there we have to make a girdle. That girdle we have to cover with usually with the help of mass plants, then different types of hormones, IAA, indole acid, like that we have to mix. Then we have to apply here. This area we have to cover with the polythene cover. After 2 to 3 months, it develops the roots. This one we have to detach from the parent plant, that will develop into the new plant if you will put in the soil. That can be seen in the pomegranate plant. So grafting method, it is divided into 4 different methods: approach grafting, cleft grafting or wedge grafting, whip grafting or slice grafting, or bud grafting. Grafting method, in this method we can introduce 2 plants belonging to same species. Desirable characters we can introduce it into the only one single plant. So here plant A, plant B, 2 plants are there. Plant A we are selecting along with the roots, that is known as rootstock. Plant B we are selecting a branch, both unite with each other, then it develops into a single plant. So afterwards we have to separate from the plant B from here. So here plant B only branch is there, whereas plant A roots are there. So that's why it is known as, whatever plant consists its own roots, that is known as rootstock. Whatever branch or twig of plant attached to the rootstock, that is known as scion. So here this plant usually exhibits plant A, plant B, both desirable characters we can see in the grafted plant. So based on the shapes they are divided into like this. So approach grafting, it is a simple method whatever we discussed. Cleft grafting, whip grafting, bud grafting, all these methods. So usually the 2 parts, that is rootstock and scion, based on the shape, incision, they are divided into like this different types.